Hey guys, I just wanted to take some time to talk to you about mitosis and meiosis. In class, we've talked about how both of these processes are responsible for cell division, and what that means is how do we make more cells? Mitosis is a very specific process done for all of our body cells, and meiosis is a process where we make gametes. And in this video, we're going to talk about the distinctions between the two. We're not going to get into full details, and you'll see what I mean when I show you these pictures, but I do want you to understand the differences and how they're similar. So first, before we can start talking about what mitosis and meiosis are, we actually need to spend some time talking about chromosomes. In our last unit, we talked about DNA. And if you don't remember, DNA is the molecular structure that has all of the information that makes us us. And if we really get down to it, DNA has the instructions to make proteins, and proteins determine everything that our body does. Now, again, DNA is super important to making us us. And another reason why it's important is because after we make more DNA, called DNA replication, we have to do something with that. And that process is known as cellular division, or mitosis and meiosis. So what we're doing in mitosis and meiosis is we're making chromosomes, and we are passing those chromosomes on to the next generation of cells. So what is a chromosome? A chromosome is a coiled structure of DNA. It is an organized structure of DNA. And so if we look at this picture here, you'll see that this looks like an X shape. Some things that I want you to be aware of, and you'll see this in your notes, chromosomes are typically only one of these lines, if you will. So like this long or this short arm and this long arm, that is one side of the chromosome. Here is the second side of the chromosome, and it's held together by this middle button looking deal known as the centromere. These two sides are genetically identical. And the reason why they're genetically identical is because we replicated DNA. So if you recall back from the last unit, let's say I have one side of DNA. Side one of DNA is ACTCGTTT. That's one side of DNA. How I would replicate the other side of DNA is I would do the opposite letters. So if my first set is ACT, my second set would be TG. So again, just replicating the second side of DNA, both identical copies, but complements of one another. So again, this is what's passed down and separated in cell division. When we make multiple cells, and I'm gonna create a picture of this for you guys tomorrow, but when we make multiple cells, the goal is to separate this so each cell, each new daughter cell gets a piece of this. So this side goes to one cell, this side goes to the other. Again, this chromosome they're only seen in cell division. So typically DNA is not beautiful and organized like this in this state. It is typically only seen like this in cell division. I want you to understand that passing genetic material is super vital. And when that genetic material is damaged in some way and you're passing that down to a cell, this is what could cause a mutation to form. So keep that in mind as we go through this the entire time. What is happening on the chromosomal level that's causing such causing wreak havoc on an organism's body? Because it all starts here at this very, very small, tiny level where one cell with one little piece of damaged DNA is allowed to mutate, and then it mutates and mutates and mutates, passing on genetic material that's not very good. So again, the process of splitting up chromosomes and passing those down to daughter cells is known as cell division. So first type of cell division, known as mitosis. First thing you'll notice is that there are a lot of things going on here. We talked about in the last video, we talked about G0, we talked about interphase. Interphase is important because remember, it does things like help the cell grow, it does DNA replication that'll eventually be used for mitosis, and then it does, again, more checking and growing. So interphase is really that good process that ensures everything is ready to go before it really gets going. So you'll see here, we spend quite a bit of time in interphase with our G1, our S, and G2. And then we go to these four little stages here known as prophase, metaphase, anaphase, and telophase. And this is the multi-phase process that splits the nucleus in half. As a reminder, the nucleus stores genetic material. When we are dividing a cell, the genetic material looks like a chromosome. So if you'll notice in this picture here, we have chromosomes. the goal is to split this up to become two. You go from one daughter cell to two daughter cells, and they are genetically identical to the parent. And this is only in mitosis. So in prophase, you'll see we have this really nice nucleus, all these organelles. And then when we get to metaphase, we see this breakdown. I am not expecting you to know what specifically happens in these four processes. What you should know is that at the start of mitosis, we have one cell. At the end of mitosis, we have two cells with two nuclei. And each one of those nuclei has genetic material. So again, we're taking all of these X's here, and I know it's a little harder to see, but if you take all these little X's, we're going to separate those X's. So if you notice in metaphase, they're lined up in the middle, almost like a great opportunity to split them in half. And then you'll see in anaphase, we split those two in half. So if you look here, it kind of looks like this process. Sorry, my dog is crying in the background. It looks like this process of splitting the two up so that one cell gets half, the other cell gets the other half. And then in telophase, we build it back up, allowing for two new cells to be formed. And then the process starts all over again. Again, a beautiful circular process. But again, the whole point of this thing is to split the nucleus in half, where genetic material is split between two daughter cells. Now, before we continue, I want you to notice this chart here. Sorry, I let my dog out, and I don't know if I'm going to edit this, so you may just heard me leave and let the dog out. I want you to notice some very particular vocabulary words, and you'll see these in your notes, the difference between a haploid cell and a diploid cell. So when we're talking about mitosis and meiosis, we are talking about two very different types of cells. And in class, we're going to spend quite a bit of time talking about homologous chromosomes. Right now, that means nothing to you. And I mentioned it at the very end of your notes, but I am going to probably have you keep an extra page at the back of your notes, which we probably will because we won't have added anything from today to tomorrow. But we are definitely going to be talking about homologous chromosomes and what makes them so special. Right now, what I want you to know is that diploid cells have two copies of each chromosome, half from mom, half from dad. So what I want you to notice about these purple sets, they're both purple, they're different shades of purple. So one of these sets, so let's say this first set is from your mom, the second set from your dad. They're the same type of chromosome. If you notice, they have the same shape, they have the same size, but they're not identical. What that means is that half of your genetics, again, comes from your mom, half the genetics comes from your dad. So if you have a mom, or we all have moms and dads, but so your mom may have, let's say, sparkling red hair, and your dad may have really, really dark black hair. They both have hair or the capability to code for hair and pass those genetics on. And then they pass that on to you. And now you have probably maybe brown hair or whatever the case may be. So again, these two chromosomes are not identical, excuse me, but they are very similar, and they happen in the fact that they have the same type of genetic material, but not identical genetic material. We're going to talk about that process of passing along homologous chromosomes in meiosis, which is on the next slide. But again, mitosis makes diploid cells. Every single cell is going to have the exact amount of chromosomes necessary for an organism to survive. For humans, that total is 46. So 46 total chromosomes, 23 pairs. The pairs come from, again, the homologous chromosomes. So 23 chromosomes come from your mom, 23 come from your dad. They come together to create 46. 46 is you. On the other side of that, we have what's known as haploid cells, and haploid cells only have one copy of chromosomes, and this is what we make in meiosis. Now, again, I just told you that, my gosh, I'm sorry, my dog is barking again. You're the worst. Okay, So, sorry. One thing I want you to notice is that on this side for haploid cells, you only have one copy of those chromosomes. And I just told you that dad gives you half, mom gives you half. So sperm, egg come together to create an organism that has a total number of diploid number of chromosomes. So in meiosis, we create gametes, and each gamete is considered a haploid cell because it has half the genetic material necessary to make an organism that would be considered diploid. But we cannot have double the amount because then the organism would have double the amount of chromosomes, and that is not good. Again, we're going to spend quite a bit of time talking about that, but I did want you to understand. Mitosis, we make diploid cells. Meiosis, we make haploid cells. And that leads me to the process of meiosis. Again, we've talked about this in class. It is a multi-phase process, very similar to mitosis. What you'll also notice about meiosis and mitosis is they have the same phase names because the process is the same, but it's not the same. What I mean by that is now we have we have double the amount of chromosomes that we need to split. So what you'll see here is we have these chromosomes. So we have 1, 2, 3, and 4, and they all replicate. They replicate to create the X shape. So remember, this side here and this side here are identical to one another. This one and this one, so this blue and this red, are the same shape, but they're not identical. They're known as homologous chromosomes. They have the similar genetic material, but it's not identical genetic material. Our goal in meiosis is to split apart homologous chromosomes. In mitosis, the goal is to just split apart the chromosomes that are already there because we've already done this homologous chromosome stuff. We're not making gametes. In mitosis, we're making body cells that already have established this is the genetic material. In meiosis, we're trying to figure out what genetic material will go on to pass on to make a baby. The other process I want you to become aware of is that in meiosis, we don't use every single cell. So not every sperm goes on to fertilize to make a baby. Not every egg that a woman drops every month is going to become a baby. So in turn, we're losing some genetic material every single time one of those sperm or egg does not fertilize. So keep that in mind, is that genetic material is different. So a woman that gets pregnant this month, and let's say she gets pregnant and unfortunately she has a miscarriage, that baby is genetically 100% different than the baby she could potentially have the next month and the next month and the next month. And this is why children look so different. And I'll use my children as an example. If you guys have ever seen, I have one child who's very much like my complexion, very tall. So he has a lot of characteristics that are similar to his dad. And again, I'll use my family as an example when we talk about genetics more. But my other son very much looks like his dad. Still tall, has genetic features, but he also has genetic features for me. So I want you to keep that in mind, is that when we're passing on this genetic material, it's a random gamble and you don't know who's getting what. But again, the whole point of meiosis is to split apart homologous chromosomes to make haploid cells. So we go through this in 2 rounds. You'll see this first round, we have. And other thing I want to point out to you is you'll notice that some of these spots have been crossed. This is called crossing over. Now, on top of splitting apart homologous chromosomes to create random gametes, we also have this process of crossing over, where pieces of homologous chromosomes swap with one another. And you don't know when it happens. It just randomly happens. And then we go on to make daughter cells daughter cells. So in this first round, homologous chromosomes are still, homologous chromosomes are separated. And then from there, we further divide each one of the chromosomes. Meiosis 2 is exactly like mitosis. The only difference in meiosis 1 is that we're splitting apart homologous chromosomes. I've said a lot and I've said this very, very fast. So I do apologize if you are super confused. We are going to spend several days going over each part of this video. So please don't panic just yet. If we don't get to our cancer lab this week, we'll just get to it next week. It's not a big deal. All right, ask me questions on a puzzle.